This week we're going to talk about cardiovascular physiology and the cardiovascular system, which includes the blood vessels, and the heart. And we're going to focus on several features. This is obviously a big topic, not only understanding the basic physiology of the cardiovascular system, but what can fail in the cardiovascular system in disease. And of course, cardiovascular disease is the number 1 or 2 cause of death in Western world, and so it's a serious and important topic for medicine. And it's also something that I realize you probably all know something about. And so we're going to try to look at things in a slightly different way, look at them the way that an engineer, a biomedical engineer, might look at the cardiovascular system, to try to understand the relationship, in particular, between pressure, blood pressure, which is the force that moves blood around through the circulatory system, and flow, the actual movement of blood, which is what's essential for distributing oxygen and nutrients and everything else that we move around in our circulatory system. So I'm going to talk briefly about the anatomy of the heart and the circulatory system. We'll start by thinking about the whole level. I'm going to talk in some detail about how pressure differences generate flow, starting with a very simple example and building up to some of the complexity that we have within our cardiovascular systems. And one of the most amazing things about the cardiovascular system is its ability to change the pattern of blood flow such that organs that need more blood can get more blood, right? It's not just that there's a static system where blood is flowing throughout the body and it flows at the same rate everywhere all the time. It increases in one region and decreases in another in response to physiological need. And we'll talk about the sort of engineering principle that the cardiovascular system uses to accomplish that. And then next week we'll talk about we'll talk in some detail this Thursday about how the heart generates the pressure that's needed to move the blood and how you can create this flow with a relatively simple but durable machine, the heart. And next Tuesday, we'll talk about cardiac conduction and electrophysiology, how the heart regulates its beating in order to generate an orderly pattern of pressure generation. So this is a cartoon of the cardiovascular system, not showing the blood except by the colors here, but showing the heart. In the center of this diagram is the heart. And surrounding it is sort of a schematic diagram like you might see for plumbing in a house, but certainly not in anatomical detail, this drawing isn't, of how blood flows from the heart to all the other regions of the body. And let's just, now I know that you've seen diagrams like this before, you understand the basic connections between things, but let's just go through them briefly. And you have to start somewhere because this is it's a closed system, right? You start at one point and you go around and you come back to the beginning again. And so if we start in this side of the heart, this is the left side of the heart. Right? And there are 2 chambers in the left side of the heart, the atrium and the ventricle. And you see that blood flows into the left side of the heart and it's come from the lungs. That blood has come from the lungs, and so it's full of oxygen, and in this diagram it's indicated as red. And if you follow the arrows here, the blood flows from the lungs into the left atrium, into the left ventricle, and out through a large vessel called the aorta. And so fairly simple so far. 2 chambers in the heart. Blood flows through these 2 chambers, atrium first, then ventricle, and out through the aorta. So if I was able to measure flow at this point here in the aorta, I would measure the total rate of flow of blood out of the heart. And you'd measure that in volume per minute, all right, in liters per minute, let's say. And all of the blood flow is coming through this one vessel. Right? And so if I could measure what the flow is through the aorta over a period of time, I would measure what's called the cardiac output, or the rate of flow that the heart is producing in its function. Now, because it's a closed system, right, the rate of flow has to be the same anywhere in the cycle. Right? If you imagined a simple tube that connects back to itself, if I'm generating 5 liters per minute of flow here, I have to have 5 liters per minute everywhere in the cycle. Right? Otherwise, what would happen? Well, otherwise, the volume of the vessels would have to be expanding or contracting at some point if the flow rate is different there, right? It would have to be changing with time if the flow rate's different. And it doesn't do that, right? And so it's a steady state system where there's a constant flow all throughout the cycle. And we're going to come back to this point, but it's important to remember. Now, immediately after the blood comes out in the aorta, there's an opportunity for it to go somewhere else. There's a branch point. And that first branch point is from the aorta into this, what's shown as a fairly large vessel, but they're pretty small vessels, actually. These are the coronary arteries, the arteries that actually bring blood to the surface of the heart in order to nourish it. And you can imagine that the heart is using a lot of energy. It's generating work. That work is being transmitted into pressure and flow of the blood, and so it gets the energy to do that from nutrients, oxygen, glucose, and so it needs a lot of blood flow. And so it's in the best possible position to get good blood flow, right? Because it's getting its blood right off of the aorta, where flow is a maximum. And we talked about stents last week that are used to treat disease in these blood vessels, and we'll talk more of that as we go through the next couple of weeks. What I want you to focus on now is just this phenomenon that the flow branches here. Right? Some of the blood goes further down the aorta, and some of it goes through the coronary arteries. Now, how much blood goes in which direction is going to be one of the main things we talk about in the rest of the lecture today. But first, let's just continue in our journey around, and you'll notice that this pattern repeats, that there are points where the flow branches. Right? For example, after the aorta, some of the flow goes up in ascending arteries. This goes up above the heart into the head, for example, and so the carotid arteries, which carry blood flow up to your brain, are descendants of this branch here, and those are important, right? Blood to the brain is pretty important, right? And also, blood goes down through the abdominal aorta, so it splits in 2 directions, right? Down the abdominal aorta, and that blood goes to your legs and all of your visceral organs that are below the heart. Right? And this pattern repeats itself, a pattern of flow, branch, 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 to ever smaller branches so that you can serve ever smaller regions of tissue. Now, these vessels that carry oxygenated blood are called arteries, and they have thick muscular walls. They're generally at large diameter, centimeter or so. There's a table in your book that gives you the diameters of different kinds of vessels. And they don't exchange materials with their outside environment. You can think of these as conduits whose function is to carry blood from the heart to a particular organ, but not to exchange nutrients, not to exchange oxygen. They're just carrying the blood. Eventually, if we looked, for example, at the branch of this artery that goes to the digestive tract, for example, to the organs of the gut, where digestion takes place, you have continual branching of these arteries until they become very fine. And those smallest conducting pathways are called arterioles. Small arteries or arterioles. Now, they have the same basic anatomy as an artery. They have a muscular wall, and we'll talk about why that's important in a minute. And they don't conduct any nutrients across their surface. They're just smaller. And eventually, those arterioles branch into very fine vessels called capillaries. And it's in these capillary beds, which are illustrated here by these little meshes, where you have the smallest diameter vessels They're feeding very particular regions of tissue, not your whole intestine, but only a very small region of the intestine. And they have thin walls that lack muscle. They're not muscular walls. And it's in these capillary beds that exchange of nutrients takes place. It's in these capillary beds that exchange of oxygen, carbon dioxide, sugar, amino acids, that takes place in capillaries. So capillaries are not muscular, but they're able to transport nutrients and molecules. Right. So what's happened as we've gone from the aorta, a single vessel, down to a capillary bed, and there are many, many capillary beds in your body. Right? Well, if I could take a cross-section at any point, a cross-section of this, then the total flow rate into all the capillaries, the total flow rate through all the capillaries has to be the same as the flow rate through the aorta. But by this process of branching, where this total flow of 5 liters per minute gets divided and divided and divided, by the time it gets to a capillary, there's a very small vessel carrying a very low flow rate, a very small fraction of this 5 milliliters per minute, but there's now millions of these vessels all over your body. Right? So the blood is not flowing at a large flow rate through a big vessel. It's flowing at a much smaller flow rate through many, many vessels. But the total has to add up to the total that goes in. Well, you know the rest of the story. The capillaries merge. After they leave the tissue area, they merge into small veins. Veins are collecting tubules for deoxygenated blood. Now, this is blood that has transported all of its nutrients into the tissue that it serves. So it doesn't have a lot of oxygen anymore. It has a lot of waste products now, the waste products that are generated by these tissues during metabolism. So it has a lot of carbon dioxide, which is the end product of glucose metabolism. Right. It has acids that are generated from metabolism. It has the end products of nitrogen metabolism, which is chiefly urea and ammonia. And that blood is indicated here as a blue line. Now, we usually show these blue-colored vessels, usually show them as red and blue, indicating lots of oxygen red, not very much oxygen blue. But it's not just oxygen that's changed, right? The chemical composition has changed dramatically. There's a lot more carbon dioxide. There's a lot more urea. There's a lot more of other things coming out of through the veins. These small veins, called venules, merge to form larger veins, eventually very large veins like the vena cava, which come back to the right side of the heart. The right side of the heart is anatomically very similar to the left side. There's an atrium, there's a ventricle, and blood flows out of the ventricle through the pulmonary artery to the lungs. Now notice that there's something different here, that on this side of the circulatory system, the part where the blood is going out to the rest of your body, the artery contains oxygenated blood. On this side, bless you, the pulmonary artery is an artery. It has the same features as a systemic artery like the aorta. It has a muscular wall, for example, and doesn't transport nutrients. So it looks like an artery, but it carries deoxygenated blood. And it's carrying it only one place, back to the lungs. And it's collected on the other side of the lungs now with oxygen and returns to the point where we started. Okay. So it's not just a closed system, it's two closed systems. Right? It's two closed systems. One that we're going to call the system that's fed by the left side of the heart, where oxygenated blood goes out to the whole periphery of your body and is collected back by the right side of your heart. That's one closed system. The second is the pulmonary vascular system, which is the force is generated by the right side of the heart. The flow is generated by the right side of the heart, flows only to the blood, to the lungs, and oxygenated blood comes back to the left side. So it's sort of like a figure 8, right? One loop, a second loop. Right? If the rate of flow is 5 liters per minute through the aorta, what's the flow rate in the pulmonary artery? They're two independent systems. Is that right? So what's the rate of flow in the pulmonary artery? Has to be 5 liters per minute because they're not independent, right? They're really, I describe them as two closed systems, really one closed system with a loop, right? With a loop. So the flow has to be the same everywhere, right? The flow has to be the same everywhere. Okay, so let's talk about blood flow in these vessels for a moment. Now you have this picture of the overall composition of the circulatory system. Let's think about a very simple problem that engineers have thought about for hundreds of years now. It turns out to have a lot of relevance in the cardiovascular system, but has relevance everywhere. It has relevance in terms of getting water to all the faucets in this building. You have to solve the same problem, and that is generating enough pressure to allow fluid to move through a length of tubing. Right? And so we'll think about a typical length of tubing, and we'll make it as simple as possible. It's a vessel that has a constant diameter or radius and a known length. And fluid is going to flow in one end of this vessel, and it's going to flow out the other. And this is called internal flow through a tube. And turns out to be one of the most well-studied problems in all of engineering. I'm just going to tell you one aspect of this, that over a certain range of flow rates, so this isn't true all the time, but it's going to be true most of the time in our circulatory system, that if I try to generate a flow through this tube, and the flow, again, is going to be measured in something like volume per minute, so let's say a liter per minute, then the pressure that I need to produce, the pressure drop, meaning what's the pressure difference from one side of the tube to the other, right? So I don't care about absolute pressure. I only care about a difference in pressure between one side and the other. That difference in pressure is proportional to the rate of flow. And that's what this equation says here. If I knew what the pressure drop was, the flow is proportional to that. And the constant of proportionality is this symbol R here, the resistance to flow. So we say that delta P, the drop in pressure over the tubing, is equal to resistance times flow rate. Delta P equals R times Q. Delta P is a driving force. Pressure difference is a driving force. It's what causes the flow. And the flow is what results from that. So delta P equals RQ. Does that kind of equation look familiar to anybody from their high school physics? Have you ever seen an equation like that? Delta P equals RQ. Driving force is equal to some constant times a flow. Right about, ever seen delta V equals RI? Anybody know where that equation comes from? Sorry? Circuits. It's the flow through an electrical circuit. What's the potential for a flow through an electrical circuit? It's a driving force. It's a voltage driving force. You hook up a battery. You hook up a battery to a length of wire, for example. What flows is electrons, current. And how much flows through a wire, right? How much flows through a circuit in response to a voltage change depends on a property of the wire, its resistance. Right? Thicker wires, more cross-section, has less resistance. You can flow more current through it. Thinner wires, less. Right? Right? Takes more, you flow for a given driving force, for a given voltage, less current flows. If you haven't seen that before, don't worry about it. If you have seen it, then hopefully this will be useful in thinking about this, which is the same thing. That pressure drop produces a flow, And how much flow you produce for a given pressure drop depends on a property called the resistance. Now, the resistance is a property of the geometry of the vessel. If I have a vessel with a very large diameter, for a given pressure drop, I can create a large flow. For a vessel with a small diameter, for that same pressure drop, I get less flow. Right? So imagine whatever your favorite beverage is. Think about a milkshake, because those are difficult to draw through straws. Right? But if you're going to drink a milkshake in your favorite flavor, do you hope you get a narrow straw or a big, thick straw? You would not pick the narrow straw if you really like the milkshake, because you can only generate a certain pressure drop with your mouth, right? And if you generate that pressure drop, you're not going to get much milkshake. You're not going to get much flow. But if I have a bigger tube, same pressure drop, bigger flow, more milkshake, right? Same thing. And here, the resistance, it turns out we can, for very simple vessels like this, the resistance can be related to the geometry of the vessel. That is, if I told you what shape this vessel was, what its diameter was, and how long it is, you can calculate what the resistance is from this formula here. R is equal to 8 mu L over pi r to the 4th. Now, you know what all these things are. You know what pi is. That's 3.14 pi is familiar. R is the radius. L is the length. What's mu? Mu is a property of the fluid, and it's the viscosity. Milkshakes have high viscosity. Water has a low viscosity. So if you're going to drink water, you're not so worried about how big your straw is. You would accept a small straw. Not just because milkshakes are more delicious, but because you don't need to generate as much of a pressure drop to move water as you do to move a milkshake, because milkshakes have a higher viscosity. The viscosity of the blood is in general a constant. It's a constant that is about 3 centipoise, and you can read in the book about units of viscosity and how they're defined, but it tells you something about the force that you need in order to cause the fluid to flow. So we're going to be dealing with fluids in here that are basically constant in their viscosity. Blood is about 3 times as viscous as water. Why is blood more viscous than water? Why is it harder to flow? It's largely water, right? It's mostly water. That's the major component. Why isn't the viscosity of blood the same as the viscosity of water? It has molecules in it, and so that makes a difference. It also has something else. 50% of the volume of blood is cells, right? And a concentrated solution of cells, like red blood cells, is not as easy to flow as water is, and so that's why water is more viscous. But if you wanted to then think about how much flow there is through this vessel, and I told you what the dimensions were, and I told you that blood was flowing, you could calculate R, and then you would know everything about flow through this vessel. Right? You would know if I have a pressure drop of so many units, this is the flow that would result. Justin? The interior surface of the tube could make a difference. Now, it turns out for low flows, surprisingly, the surface of the tube doesn't make a difference if the flow rate is below a certain level. And you're thinking about, what if the surface of the tube is rough? Then fluid that's flowing past that surface is going to experience more friction than if it was flowing past a very smooth surface. At low flow rates, the friction that occurs when a liquid meets a solid is so high that fluid at the surface doesn't really move at all. For low flow rates, fluid that's right at the surface doesn't move at all, and so roughness doesn't matter when you have no flow there. But as the flow rate increases, then roughness becomes more important. So 2 things about it in our vascular systems. The flow rates are generally low enough that you don't have to worry about that. And our vessels are all really very similar in terms of their microscopic geometry, and that all of the blood vessels are covered with a monolayer of cells called endothelial cells that basically form a blanket over the entire surface of the inside of the circulatory system. And so there's very little frictional resistance to flow there. But it's a really good point. Other questions? Okay. So this equation sort of makes sense, right? If you go on to study biomedical engineering, you'll learn exactly where this equation comes from, and you'd be able to derive it yourself. But for now, just accept it. And it is true for almost all the vessels in our body, almost under all the circumstances of human physiology. And it tells us that resistance varies with viscosity, length, and radius. And it varies in kind of the way that you would expect. As viscosity of the fluid goes up, resistance goes up. As the length of the tube goes up, resistance goes up, right? Harder to pull through a long straw than through a short straw. And as the radius goes up, resistance goes down, right? Because the radius is in the denominator here. So as radius goes up, the resistance goes down. And remarkably, the resistance goes with the fourth power of the radius. What does that mean? That means a twofold increase in radius leads to a 16-fold reduction. 16 is 2 to the fourth, right? A 16-fold reduction in resistance. So resistance is, of all the parameters, resistance is most sensitive to radius. Caitlin, did you have a question? Yeah. So what's the unit here? Well, let's think of it. What unit would you measure pressure in? What's a unit of pressure? Pascals is one. Others? Newtons per meter squared or millimeters squared. So it's force per area is pressure. What's a more commonly? Those are great units. Sorry? Pounds per square inch is another unit, PSI. What's the pressure in the room here? 1 atmosphere. Atmospheres is another pressure, so relative to atmospheric pressure. And if you have any others? Millimeters of mercury. It turns out that all of those are proper units for pressure, and you could convert between one or the other as long as you knew what the conversion factor was between the. We usually use atmospheres to describe atmospheric pressure. Physiologists usually use millimeters of mercury to describe pressure in the circulatory system. Millimeters of mercury. So 1 atmosphere is 760 millimeters of mercury. What are the blood pressures that are relevant in your circulatory system? How many millimeters of mercury do you think? Anybody know what their blood pressure is? (120/80) what? Don't tell you that. Right? 120 millimeters of mercury over 80 millimeters of mercury. And we're going to talk about where those numbers are, where those pressures actually exist in a few minutes. But that's the range of pressures in your circulatory system, from 0 millimeters of mercury up to 120 millimeters of mercury, roughly. And we'll see you go outside of that range sometimes. Now, these are pressures that are inside our circulatory system, so they must be, but they're only a fraction of an atmosphere. How can that be? Right? An atmosphere is 760 millimeters of mercury. That's the pressure in this room. If your blood pressure is 120/80, what does that mean? Well, pressure is we only care about differences in pressure. It's relative pressure that matters, not the absolute pressure. So when you say that your blood pressure is 120/80, that means 120 millimeters of mercury above atmospheric, 80 millimeters of mercury above atmospheric. Right? Otherwise, our blood vessels would collapse because we're surrounded by air that's 760 millimeters of mercury. That to inflate those vessels, you have to be at pressures above that. But since all we care about are pressure differences, then the pressure that you measure when you measure blood pressure is pressure above atmospheric. Does it make sense? So delta P is in millimeters of mercury. Q is in liters per minute. So R is in units of millimeters of mercury per liter per minute. Or millimeters, right? It's in units of, if we just write this out, millimeters of mercury equals R, and this is liters per minute, then R must be in units of millimeters of mercury per liter per minute. Okay. So this resistance changes. It changes with geometry. The most sensitive thing that changes is the radius. And so imagine that if you had a vessel that could change its radius, that could become smaller or could become larger, right? Then that would be a vessel that could regulate its resistance. Right? And so if that vessel was faced with a pressure drop, let's say a pressure drop of 10 millimeters of mercury from beginning to end of this vessel here, and it could change its radius, then it could change the amount of blood flow through it. If it decreased its radius, the rate of blood flow would go down, even though the pressure drop stays the same. And if it expanded its radius, the rate of flow would go up, even though the pressure drop remains the same. Does that make sense? So I mentioned before that arteries, arterioles, small arteries, all arteries have muscular walls. And they have a special kind of muscle called smooth muscle, which is arranged circumferentially, sort of wrapped around the vessel. And that muscle can contract to constrict the vessel or relax to dilate the vessel. And so one feature of arteries that makes them important in physiology is that they can change their diameter. And when they change their diameter, they change their resistance, and they change the flow that goes through them at a fixed pressure drop. So why is that important? Well, let's say you get up and you start running, right? The lecture's over. You're excited to tell your friends about it. So you get up and you run out of class. So you went from resting to running. Now all of a sudden your muscles need more blood, because they're going to start generating work. And so the arteries that feed your muscles are going to contract, are going to relax. Diameter will get bigger. The pressure available is the same. They'll get more flow. That flow has to come from somewhere, so simultaneously other vessels are constricting, right? And what would those vessels be? Well, probably the vessels that go to non-essential functions when you're starting to run, not your brain. You want to keep blood flow to your brain so you don't run into the wall, right? But you might have less flow going to your digestive organs. So you'd stop digestion for some period or slow it down, provide less blood flow for that purpose. Does that make sense? Well, if we look at how pressure varies through the circulatory system, your blood pressure is generated by the heart. And blood pressure ranges between 120 and 80 millimeters of mercury. And we're going to see this in a few minutes, but those numbers represent the maximum pressure and the minimum pressure during a heartbeat. The maximum pressure occurs when the heart is actively contracting or beating. We'll talk about that more in a few minutes, and we'll talk about that in great detail on Thursday. And then it drops when the heart is relaxing. So blood pressure is generated in a cyclic fashion because of beating of the heart. Contract, relax, 120, 80, 120, 80, 120, 80. And that pressure of 120, 80 gets transmitted to the aorta. And the pressure in the aorta is roughly the same as it is in the heart. Now, why is that? Why isn't there a pressure drop through the aorta? Well, there is a pressure drop through the aorta, but it's very small. And the reason it's small is that the aorta is big, has a large diameter, and so its resistance is low. Its resistance is low, and so in order to generate 5 liters per minute of flow, you don't need much pressure. In order to even this whopping flow that goes through the aorta, because of the large size of the aorta, you don't need much pressure drop to generate that flow. So if I measured pressure drop over the aorta, from the beginning to the end of the aorta, I'd find that pressure doesn't really change very much, and that's what's shown in this diagram here. As I move from the beginning of the aorta to the end, there must be some drop in pressure. Has to be. Otherwise, blood wouldn't flow at all. But there's not very much of a drop. Similarly, the large arteries, which still have large diameters, have low resistances, doesn't take much pressure to move a flow rate through those large arteries. So over the large arteries, there's not much pressure drop either. So this would be the branches, like the main branch that goes to your arm, the branch that goes, the carotids that goes up to your brain, the branch that goes down your leg called the femoral artery. Still large arteries, large diameter, not much pressure drop needed to generate a large flow. As we move to more branches, smaller and smaller vessels, it turns out at some point, and the point at which this occurs is the arterioles, the diameter has gotten small enough that the resistance becomes significant compared to the flow rate. The resistance becomes significant compared to the flow rate. Now, there's so many branches that occurred before this. There's so many branches that these individual vessels aren't getting 5 liters per minute, right? They're only getting some small fraction of that. There might be 1,000 of these arterioles, let's say. So they're all getting 1/1000th of the maximum flow. So how could that pressure drop be more? How come they need a big pressure drop, which is what's illustrated here? It takes a substantial drop in pressure in order to move this lower flow rate through these smaller vessels. How could that be? Well, remember that the resistance changed with the radius to the 4th power. And that means that as the radius goes down, it's not a linear relationship between the radius going down and the resistance going up. It's a 4th power relationship. So as it gets small, resistance starts to become very, very high. When you divide a flow between vessels, that's kind of a linear process, right? It goes 2 ways, it goes 4 ways, it goes 8, but this radius to the 4th power starts to dominate when you get down to a certain radius. And it turns out that that's at the level of the arterioles. So most of the pressure drop on this side of on this left side of the cardiovascular system, most of the pressure drop occurs at the level of the arterioles. There's a significant pressure drop through the arteries, and then not much pressure drop at all through the veins. The veins are low-pressure vessels, generally large in diameter, and not much resistance to flow. Now, why, if you were going to design a system like this, is there any advantage to having the largest pressure drop occur over the arterioles, or the largest relative resistance occurring over the arterioles? Well, there's a huge advantage, because wherever the pressure drop occurs, that's the place where you have an opportunity to adjust the flow. That's the place where you have the best opportunity to adjust the flow, because small changes in radius are going to lead to large changes in relative resistance. Right? So it's at the arterioles, or the very smallest arteries that are supplying blood to our tissues, where we have the most control over flow rate. And that makes sense, because sometimes you're not making big movements in blood flow, like saying I want very little blood flow to my gut and a lot of blood flow to my muscle. That's a big change. You're making smaller changes within a tissue, like I want more blood to go to the region of my brain that's involved in talking, and less to the region of my brain that's involved in reading. Right? When you're talking versus, right? You want to regulate where the blood flows on a more smaller scale, on the scale of a tissue. And it's arterioles, which are branching within tissues, that have the capacity to do that. So that you could do something like this. And let's say this is a very imaginary situation, but I drew it just for illustrative purposes. Let's say that this was an artery that was feeding some tissue, and so there was a blood flow that's coming in here, and it splits 5 different ways. And so here are 5 different regions of the tissue that are served by these arterioles. Now, I could draw this. One of the nice things, if you do know this analogy between this equation describing pressure drop flow relationships and this equation describing voltage current relationships, is that you could draw a diagram for pressure flow relationships. It looks just like a circuit diagram because the equations are the same. And that's what I've done here, is taken this anatomical picture, cartoon, and converted it into an equivalent circuit, where I'm not thinking about flow of electrons now, or current. I'm thinking about flow of fluid. But I'm drawing a resistance not as an electrical resistance, but as a flow resistance, as this kind of a resistance. Right? And let's say that the blood is flowing in at a rate of 100 milliliters per minute, and that the resistance here is 1 unit, and the resistance here on each of these smaller segments is smaller, or higher, right? These are smaller. It branches into smaller vessels, so they have a higher resistance. And it's 5 units here and 1 unit here, and the units are these. Well, if all the resistances are the same, then of the blood that flows in here, it's going to flow equally through each one of these tubes. It's going to flow equally through each one of those tubes, and so basically this 100 milliliters per minute of flow gets split 5 ways. Right? You could do this calculation yourself. The pressure drop here is just 1 millimeter of mercury, and if you did this calculation, it should work out okay. The total resistance of this circuit with 1 millimeter of mercury gives you 100 milliliters per minute of flow. Okay. What if one of these vessels changes its diameter? One of these vessels changes its diameter. Let's say it's this one, and let's say that it shrinks its diameter so that its resistance goes up. Goes up here from 5 units to 100 units, so I made a big change in that. Right? It goes up by a factor of 20, so that would be about a factor of 2 difference in radius because of the 4th power. Right? If this one goes up to 100, now there's more resistance through this particular vessel here, and the same resistance through these 3. So what happens? Less flow goes through this vessel, and the same flow goes through the others. So that the total flow now is 89 milliliters per minute. Now, why did the total, you could understand, sort of, because that's pretty obvious, right, why when I decreased the radius and increased the resistance of this vessel, why less flow went through it than through its partners. That kind of makes sense. I've got a smaller vessel, so now I've got 4 large vessels and 1 smaller one, and if I feed them with the same pressure drop, more of the flow is going to go through the big vessels because there's least resistance there. Fluid takes the path of least resistance. Some goes through, but not as much. Right? That makes sense. But why does the overall flow through the whole circuit change? Why does the overall flow through this whole thing change? Well, because if any one of the vessels in here increases its resistance, it increases the overall resistance of the aggregate of vessels, right? And if the pressure that I have available is constant, and it is, the pressure I have available is constant. In this example, it's 1 millimeter of mercury. Then when the whole thing increases its resistance, that means the flow rate through the whole circuit has to go down as well. So there's 2 things that are really illustrated here. One is that you could regulate in pathways that are competing with each other for flow. Then the relative resistance between equal members determines how much flow goes to each vessel. Right? That's one thing that's illustrated. The other is that the flow through this whole piece of the circulatory system, this whole part of the network, depends on the resistance of every single vessel within it. And so one vessel changing its resistance changes the whole thing. And that's a feature of our circulatory system, right? If I want more blood to go to the brain, there's 2 ways to get more blood to the brain. Well, there's more than 2 ways, but 2 ways as long as the blood pressure stays the same. One way is to dilate the vessels of the brain to create less resistance there. The other way is to constrict the vessels somewhere else to create more resistance there. Either one of those is going to get me more flow to the brain. And so it's because it's a closed system, everything is interconnected. and a change in flow in one place can influence flows throughout the whole system. Does this make sense? I want to go back to this picture now and talk about what we've learned in terms of flows. And now you can think about this in a little bit different way, that if there's flows going through here and here, how much goes each way, each direction, depends on the relative resistance between this vessel and this vessel. Yes? It does depend on the relative size between this vessel and this vessel, but it depends on the relative resistance of the whole network after it as well. It depends on the whole resistance. Even if you have a big tube here, if it's feeding lots of very constricted tubes down here, the overall resistance that the fluid experiences at this branch point is going to be high in this direction. Right? The resistance of the whole circuit matters. So think about that. And then let's now think about what's driving this flow. That is, what's generating the pressure. And you know that what's generating the pressure is the heart. That's the only place where pressure gets generated within this closed system. And the pressure that's generated by the heart, which is experienced by the aorta, is what drives flow throughout all of these vessels. And the only opportunity that individual vessels out here have to adjust how much flow goes to them is by changing their diameters, which they do. Right? The only way to change the overall rate of flow is to change the overall pressure drop, which can only be done by the heart. How does the heart work? Well, let me just start this, and then we'll talk about this in much more detail next time. This is a picture of the heart. It sits about the size of your fist, basically in the center of your chest, but shifted a little bit to the left-hand side. You can find out where yours are by just putting a finger there and feeling where you can feel the heartbeat. It's fairly easy to define. The apex of the heart, the tip of it sits downward, like that, and the major vessels are near the center line. So the tip is down here, the major vessels are near the center line. Right? The heart has muscular walls. The walls of the heart are called the myocardium, muscle heart. Myo is muscle, cardium is heart. And there's muscular walls. So it's a big muscle, basically. And there are blood vessels on the surface. And so this is the aorta coming up here. Where this arrow is here is the aorta. This is the aortic arch, and then goes down to the abdomen and up to the brain. And these are those coronary vessels that I mentioned earlier. The first branches off of the aorta are to these coronary arteries, which branch over the surface of the heart. So now you can see this branching pattern more clearly in one particular tissue. Here's a coronary artery that blood flows through. Here's a branch. One goes more to the left. One goes more down the center line. Branches again, and by continuously branching, you define smaller arteries which serve smaller regions of tissue. These are the blood vessels provide nutrients to the muscle in the muscular walls of the heart. That there are inflows and outflows. You know this is the aorta up here. This is the vena cava, which is bringing blood back down here. The heart has a right side and a left side. If I divided it into the right side and the left side, I could put a dotted line here where the left ventricle and left atrium are on this side of the line, and the right ventricle and the right atrium are on the other side of the line. And what we'll see when we talk about this more next time is that the left side of the heart is more muscular and slightly larger than the right side of the heart, even though they're generating the same flow rate. Right? The left side of the heart is larger. If we reduce this down to a simpler picture, this is where I'm going to start next time, and take out the anatomy and replace it with this cartoon, you can see that there's a difference between the thickness of the muscular walls. The muscular walls on the left side of the heart are slightly